I can give you a quick welcome
Sure.
for everyone who's joining this morning. It's still morning for us. I don't know about everyone else. We're going into the section where we're going to go through metabolic modeling in KBase. And so we have Janaka and later Chris will be joining us to take us through how you use KBase.
Ready. Thank you, Leanne, and welcome back, everyone. In a Friday afternoon, joining us to listen to our presentations and the metabolic modeling tools in KBase. So really quick, I will go to show the KBase page, kbase.us. And if you are new to KBase, just please go here and sign up for a new account. It takes a minute. And you can use your Google ID, Oak ID, or if you have a Globus ID, you can use that to create an account. And this is super useful because later today that we'll go through the workflows that what we are discussing with the students. And this is also open to the public. And once you have an account, we have set up an organization called EMSL 2021 Summer School. Once you log in to the KBase, you can go to this org section and just select all orgs and type EMSL 2021. I have a chunk of stuff here, but if you are new to KBase, you probably have only this organization or very few organizations that listed here. Go here into the organization, and there should be a button for you that says join this organization. And once you join, we can approve that. So you will have the access to all the workflows, the workflow that we're discussing today, which is the EMSL Summer School 2021 Day 5, and also other useful tutorial narratives, and also the 2020 PNNL EMSL Summer School workflows. So it's very useful, and just take a couple of minutes to create an account. So with that, I will go into my presentation, and I'm going to close my video just to save some bandwidth. Been having some network issues this morning. Let me. Can you see my full screen?
Yes.
Yeah.
Already. So this presentation, I'm going to talk about the broad overview of metabolic modeling and reconstruction analysis tools in KBase. As we have seen, the participants are from diverse backgrounds, from computationally savvy to experimentalists. So and broad overview would help on what we do, on what kind of tools that we have available on KBase for metabolic modeling. First of all, acknowledgements for TAs that this afternoon that will guide students in the breakout sessions. Thank you to all. So we have seen that Jeremy talked about in-depth influx balance analysis this morning, but let's go in very broadly, like why we need metabolic models, what are metabolic models, how the metabolic models made, and what kind of tools out there to use it for, especially for people that are not computer savvy. How can you use these powerful tools for your analysis and answer your biological questions? So we know that there are organisms that these days, there are tons of organisms get sequenced in very little time, and there's tons of sequencing data get accumulated. So we run through this sequencing data through assemblers, and then we run this data through annotation pipelines. Once the genomes are annotated, you will see like structural annotations and functional annotations. So the structural annotations are the protein sequences, and the functional annotation, and the annotation algorithms assign a function like pyruvate synthase or NADP transhydrogenase. So we have an idea what this genome consists of, what kind of functions this genome has. But if we want to study the behavior of the organism, can this organism grow on certain media formulation? Can it grow in a minimal media? Can it grow aerobically versus anaerobically? Can it would be able to produce an important biofuel compound. So if you want to answer that kind of questions or to study the behavior, metabolic models, among other tools, are one of the powerful tools that you could use to derive these conclusions. So we use different biochemistry databases like KEGG and other KEGG, Metaseg, and other biochemistry databases to derive these reactions, which I'm going to talk in a minute, and build these metabolic models. So in this context, so we could build metabolic models right now in KBase for bacteria. You can build metabolic models for fungal and plants, and then you can use these metabolic models to study the environmental systems. Like, if your environmental system consists of, let's say, plant root system and a bacteria, or fungal and bacteria, you can model these bacterial system as a compartmentalized metabolic models and try to identify what kind of a overall metabolic capability that these organisms have, what kind of excretion products that these organisms could produce, how they interact between each of these systems. In this context, I added this slide just before my presentation, seeing some of the questions in the Discord and on the Zoom chat. So the KBase has end-to-end pipelines that users can use sequencing reads for microbiomes, and they can assemble microbiomes. They can bin to extract genomes from these microbiomes, and then you can model the entire microbiome in KBase and also the isolated bins, and see and try to analyze these these bin models or the entire microbiome models. So we do have tools to build metagenome models. So if we want to answer questions like, what is the overall metabolic capability in this microbiome? Is this microbiome could produce a certain biomass compound? To answer that kind of questions, you could use entire microbiome models. And if you want to ask more specific, okay, what are the organisms in this microbiome could fix carbon dioxide, could reduce nitrate, and what kind of compounds that they could excrete that some organism could use as a carbon source. For an instance, if one organism could fix carbon dioxide into complex carbon molecule and excrete acetate, another organism can use acetate as a carbon source. So that if you want to derive that kind of inferences, so a compartmentalized community models will be a great tool. And we have in KBase, we have the tools to build compartmentalized community models. So it's a very important talk. What can metabolic models do in general? So as I mentioned earlier, so it could be used for in community model context that identify trophic interactions among organisms and predict impact of genetic perturbations. So if some organisms are not culturable in the laboratory conditions, and if you want to identify a minimal media composition that these organisms can be cultured, you could use the help of modeling to identify a list of compounds for that. Can we optimize a certain biofuel product like ethanol? So in today's workflow that we are going to present, so we have an example that how you optimize the production of ethanol in yeast, for an instance. So and also predict culture conditions and possible responses to environmental changes. So these in silico simulations are very useful to derive this kind of conclusions. And then once you know like the culture media conditions, then you have a head start to go to the laboratory and start and use those predictions and to answer your biological questions. So in silico simulations, when we have metabolic models and when we do in silico simulations, there's a certain accuracy rate, and this accuracy rate computationally it's not going to be very, very high. So where we can improve the accuracy rate is integrate the laboratory generated phenotypic data, multi-omics data integration. So if we can integrate the metabolomics that you have heard a lot of information about the metabolomics, the proteomics, and also the transcriptomics. And these are the measurements that was produced from the laboratories. And if we can integrate these measurements into the models, your accuracy rates are very high, and you get much more realistic simulations with the reconciled phenotypic data. And this afternoon, Chris going to talk about the metabolomics and how the metabolomics get integrated into the models in depth. And also this, this interesting area of cheminformatics, that how you can expand from the known biochemistry and producing these novel compounds computationally, and then try to map these novel compounds into the metabolomics data. So on day 3, I think that I heard this Natalie's presentation, that even for well-known organisms like E. coli for prokaryotic, so model organism, and for yeast for eukaryotic model organisms, there's about 2,000 to 17,000. I could be wrong on the number. There's tons of metabolites that are still unknown. So we know little still. So the cheminformatics is a great area to see that, okay, what are these novel metabolites that we don't know, how we can map those novel metabolites into these computationally derived novel compounds. And then we can integrate those novel compounds into metabolic models, and then we can see how they play a role in that organism's behavior. So in omics integration, in KBase, there are tools like I'm showing here a couple of examples that you can visualize omics data, like the transcriptomic data and metabolomics data and fluxes in Asia-based pathway maps. So what you're seeing here is like, I have the TCA cycle around here, and then the aromatic amino acid biosynthesis. And the green color shows the fluxes or expression. So you can paint the fluxes or expression on based on a flux distribution in a pathway map. And you can also show the abundance of metabolites in the pathway map. And I've seen that Jeremy showed some of this in earlier presentations. So you can do this in KBase, that you don't have to worry about installing any dependencies. You don't have to worry about installing any software. Anybody with zero or very little computational experience, but running a simple app, that you can do this kind of analysis and see what are the high flux pathways, and is the fluxes and the expression analysis make sense? Do you have the high expression in high flux pathways? So that kind of analysis. So this is another example that I'm integrating transcriptomic data from yeast to an yeast model. So we built a model that this is also part of the workflow that you can see in the narrative, and load transcriptomic data sets, and then you compare gene expression and analysis with the flux predictions. What you saw here is that if you have the red, which is the active flux and active expression, and then the brown is no flux and no expression, so how a model agree or disagree compared to the expression analysis, compared to the expression levels. So what you see on the y-axis here is the metabolic pathways. These are the KEGG metabolic pathways, and you see if you see lots of red and brown, that means shows the agreement, because that shows the active flux and expression, and that's the brown shows the no flux and no expression. So here, based on the flux distribution, you see that how to what extent a metabolic model can agree or disagree. If you have lots of brown and red, that means good. If you have lots of tan, that means that's not good. So that means that either there are fluxes, but there's no expression, or vice versa. So the blue represents that there is gap filling. There's lots of gap filling. That means in that metabolic network, there's lots of gaps, and an algorithm has to fill those gaps, which I'm going to talk in a second about the gap filling. So let's now take a look at it. What is a metabolic model? And so the metabolic model is a list of all reactions involved in the metabolic pathways, and a list of roles associating reaction activity to gene activity, and a biomass reaction listing essential building blocks needed for growth and division. So we have a metabolic network and we have a biomass. So in modeling terminology, usually we use the term objective function, and typically the objective function that you hear most of the time is biomass, because many of the folks that use models want to measure the growth. When you measure the growth, the objective function is biomass, and the biomass is experimentally derived, and so that consists of building blocks of the cell, like the amino acid, nucleotides, lipids, cofactors, cell wall components, energy, the quinones, and that kind of stuff. So the objective function not necessarily needs to be biomass. It can be an excretion reaction. So the objective is to optimize the production of a certain biofuel compound, and but typically it's the biomass, because people want to measure the growth. And the way we derive reactions into this metabolic network is you have genes and you have functions that are associated with genes, and genes transcribed and translated into enzymes, and from these enzymes we mapped to biochemical reactions based on the biochemistry databases, and then we add those to the model. And also, to simulate a model, you need a media formulation like glucose minimal media. So if it's or in this example, if it's glucose minimal media, it uses the glucose as the sole carbon source, and it tries to produce all these biomass compound from that sole carbon source. So in KBase, we have pipelines to construct models for bacteria, for fungal, and for plant, and they use different strategies to produce metabolic models. So for bacteria, we have annotation pipelines like RAST that annotate bacterial assemblies and assign functional roles like pyruvate kinase or NADP transhydrogenase. And we map these functional roles to specific biochemical reactions. And these are like a control vocabulary that we map these functional roles to these biochemical reactions. In addition to that, recently we added other ontologies in addition to RAST annotations, like EC numbers, like KKOs, like Go terms, that we could build metabolic models from these annotations in KBase. And for fungal, and also the plant, the strategies that we have a curated set of biochemistry that is derived from published metabolic models. And the way we propagate that biochemistry into the genome of interest is that we use we compute orthologous protein families between these published models and of your genome of interest, and then we propagate the biochemistry based on those orthologous protein families. So since Jeremy talks FBA in depth, so I'm not going to go into deep, but in very, very broadly. So the FBA is flux balance analysis, and it's a mathematical approach for analyzing the flow of metabolites through a metabolic network. So if you see in this toy cartoonish model, that what you see is that I have a toy model that I have these compounds and reactions. Now, if you imagine that a compounds come into the cell at a certain rate, let's say the rate of 60 here. So when you see this, just imagine that you can visualize like in your mind that this is a linear pathway like glycolysis, that glucose coming into the cell and it makes pyruvate. So the type of models that we use is we call steady state models. So the steady state, by definition, that means there's no internal metabolites allowed to accumulate nor degrade. That means if this compound come into the cell and change to another compound through an enzyme at the rate of 60, you would see that the same rates towards producing the end product. So the internal compounds cannot degrade or allowed to accumulate. So the conversion rate has to be consistent, and in order for this steady state modeling to work. Now let's take a look at slightly more complicated model that here that we have 3 separate routes that are equally valid solutions and equally independent, and if we still consider that a compound coming in like glucose at the rate of 60, and this can now divide into 30, 20, and 10, but still if you add these values, still it's going to be 60. So that's what I meant by steady state, that compounds cannot be degraded nor accumulated. So always the rate has to be balanced. So now one of the big problems that we have in steady state models is that although the condition is that the compounds cannot accumulate nor degrade, but the reactions, some of the reactions are missing in this network. This is due to annotation gaps. This is due to the gaps in our knowledge. We don't know what those reactions are. This is even true for E. coli or like yeast, probably the most well-studied organisms for prokaryotes and eukaryotes. So because of this problem, we have metabolic gaps in networks. So in order to use these models, we need to fill these gaps. So let's see how what's the problem is, like as I mentioned earlier. So if we have this reaction is missing in this network, this system doesn't work. So we need to somehow fill this gap. And how do we fill these gaps? So people have different strategies filling these gaps. So right now in KBase, by default, you try to use a minimal set of reactions to fill these gaps, and there are other algorithms that people want to use fill these gaps that like biologically more sounds, they want to see that what are the reactions that you can find for that organisms that are phylogenetically closely related to that organisms. Can you find a solution from another organisms that phylogenetically close or metabolically close, and then add those reactions? So there are different strategies. So let's take a couple of scenarios that how we gap fill a metabolic model. The couple of scenarios that what I have here is that the gap filling is entirely dependent on its medium. So I'm giving these scenarios like how we gap fill in rich medium versus minimal medium. And so this is my slightly more complicated than the model earlier. So we have these biomass compounds around, and we have a reaction network, and we have metabolic gaps. Now, if you try to gap fill in rich medium, so the rich medium is like LB. It has all kinds of nutrients in it, amino acids, all kinds of vitamins, quinones, lots of nutrients are there. So if you try to grow an organism in the laboratory on LB, it doesn't need to fire up all the pathways to synthesize the end compounds, because it can simply uptake those nutrients from the media like amino acids and vitamins and try to produce this biomass. So that's exactly happens when you try to simulate a metabolic model and when you try to gap fill in rich medium. So in rich medium, all these compounds are given in the media formulation. So what the model does, it simply uptake these compounds into the model and satisfy the biomass. So any of these gaps inside the cell that to biosynthesize wouldn't be filled, wouldn't be activated. Now, this case is different if you try to gap fill in minimal media. Now, let's take the scenario that you try to gap fill in glucose minimal media. So in that case, the sole carbon source is the glucose, that it has to produce all these biomass compounds based on the glucose minimal media. In that case, it will gap fill all these biosynthetic pathways in order to produce these compounds. So if you want to get a model that are fairly more complete, a functional model, and it's the organism can grow on minimal media, so you want to gap fill in minimal media. And as I mentioned earlier, so these reactions come from biochemistry databases like KEGG, MetaSeq, and BIG, and published models, and that's how we derive these reactions. We maintain a model seed repository that all these reconciled biochemistry reaction in model seed ontology, and the KBase currently use model seed ontology to represent the reactions and compounds in KBase. That everything I explained so far that you can do just by running apps. You don't need to have lots of computer knowledge to run these apps. So that's with that. I have one more slide that I want to show. That I want to first acknowledge the KBase team and also the help from the EMSL team to for this to get this workshop going, and all the help to for us to give these presentations. And so I was telling that to run all these apps that I was showing earlier, you don't need you need very little computer knowledge. You don't need computational knowledge at all. And if you are an experimentalist, if you are a biologist, you want to run these apps, that you can run these apps. So in KBase, all these apps are just select the input and a push of a button that you can run this app and get your analysis. So I have about, Becky, how much time I have?
It is 10:57 right now.
And my presentation ends at 15 minutes.
Until 11:15.
Okay, great.
So you have over 15 minutes.
Okay, great. So now I'm going to KBase, and if you already went here to this organization and request for an access, we would approve that right away, and you will have the access to these workflows. I'm using this EMSL Summer School 2001 Day 5 narrative. And so I'm going into this narrative. So what you see here is that in a environment that you have all your data objects, that you have your workflows, that you have commentary, everything in one place, how this workflow is being run. And you can share this workflow, the data, everything in a click of a button with your group, with your students, and it run the same workflow with your own data. So there's lots of advantages doing in this kind of an environment. So I'm going to just walk through really quick with the available time, and we will go through this work with this workflow with the students this afternoon in depth and answer their questions. But if you have questions, just please type in the Zoom chat or in the Discord chat, and we have my colleagues from KBase that can answer those questions. So what we are doing here is that this is a narrative that we import a yeast genome into the narrative, and we construct a draft metabolic model, and then we gap fill that metabolic model. We run flux balance analysis. We upload omics data into the narrative, the transcriptomic data and the metabolomics data, and we integrate those data into the metabolic model. We visualize these transcriptomic and metabolomics and fluxes in the base in the pathway maps to see which path we have fluxes. We also have an example that how you can engineer an organisms that if you can compute a certain set of reactions or genes to knock out, can you optimize a certain biofuel product like ethanol. So just walk through here that you can copy a genome into the narrative. So in a narrative, if you go here and into the public tab, there are tons of public genomes are available that you can copy into the narrative. So right now it's about over 234,000 genomes available in bacterial, archaeal, eukaryotic genomes that you can copy into the narrative. And so in this case, I have imported a genome from outside. You can import any genome into the KBase from your local machine. And if you go to the import tab and drag and drop, you have instructions here. And you can also access the genome that I have used here and import it into narrative. And you can browse the genomes, what kind of features that you have within the narrative interface, and access to the genome sequences, the protein sequences, and so forth. And then we constructed a draft metabolic model of yeast. So here we use the build fungal model lab. So this is what I was talking earlier. So you just select the genome that you want, and many of these are very, very standard. So there are advanced options that you can read documentation and what those advanced options does. But basically, you just hit the run button here, and that will generate a draft metabolic model. That's easy as that. And this will also give an analysis of how will this compare to other published models, what kind of reactions, and the amount of reactions, the composition of reactions that comes from the other matched to other published models, known published models that was produced for fungi. And then the model is made, so it will show all the reactions that you can have access in a table format, the reactions in the model, and also like many of the reactions are associated with its genes. So this is very important. So you can see which genes propagated what reactions, and also gives the biochemical equation, the reaction name, and also it gives a list of compounds, a list of genes, and also the reactions again here. And also it will give this compartment. So if you can recall that I was talking about the compartments during my presentation. So eukaryotic models, unlike prokaryotes, have organelles. So each organelle is represented in its compartment. And this is the biomass equation. This is the biomass that I and Jeremy was talking earlier. So you can have access to all the compounds that the biomass is made of. And if there are any gap filling that we have done for these models, and also it shows the pathways. And right now we have the KEGG pathways that listed here. So if you click on any KEGG map, it will paint the reactions that are present in this model. And it will also that we have the commentary that what each compartment means. And then we are doing a we are imported a media from the KBase public data here. If you go here, and then if you go to this drop down, you can see the other sources that where you can import the genomes and also the media. Like in this case, I selected the media. And if I just type carbon the glucose, for instance, I can select glucose minimal media and then add to the narrative. And so that's what I've done here. And you could see the media compounds that what's in here in human readable format. And then we imported this media because we want to now gap fill the model in minimal media and then run flux balance analysis. I also have guiding questions for users and also for our students this afternoon. And so in this case, we have an app gap fill metabolic model, which fill the gaps in draft metabolic models. In this case, we are using the glucose minimal media to gap fill this network. And then it will give an output, the reactions that, what are the reactions that was gap filled, and what are the reactions that been altered, like change the directions during the gap filling in order to get a valid flux solution. Then we run flux balance analysis here that I have literature articles related to the FBA and flux variability analysis that Jeremy was talking earlier. And you can get lots of information, and there's tons of videos in YouTube if you want to study FBA, FVA, and in general model metabolic model construction. So here we run the flux balance analysis based on the glucose minimal media. And we use the app run flux balance analysis. You select the model that you constructed above, and you select your media that you want to run flux balance analysis. In other words, if you want to grow this yeast model in glucose, you just select that media and you run the flux balance analysis, and then you get a flux distribution. So in this output table, now you could see the fluxes by those reactions. So if I sort this table, so you can get a list of reactions that have fluxes. And it also shows the exchange fluxes. So here, let's check the the ethanol. Does it produce any ethanol? And you can see that it's produced. The negative means it's producing, so it's excreting out and some ethanol in the when the organisms grows aerobically. And you could see that kind of a data in this table format by exploring it. And then you can grow anaerobically. So most of the time, to get fermentation products, you grow these organisms anaerobically, right? So that should reflect when you try to simulate anaerobically. And if you simulate anaerobically, and then you get another flux profile, and understandably, I could see the objective value of the growth went down compared to aerobic FBA. And now if you check the ethanol production in exchange fluxes, and it went higher. So that makes sense. In anaerobic growth, there should be more fermentation and the flux move towards the producing ethanol. And can we optimize the production of ethanol using an engineering technique? So there are tools like OptGene that you can find all about it through this literature. And if you have certain genes knocked out, or in this case, the reactions knocked out, you can get increased amount of ethanol. And in this case, what we are demonstrating is that previously you saw that there's a small amount of ethanol being excreted when organisms grows aerobically. And I'm still growing the organisms aerobically, but now I'm knocking out this set of reactions. And with that knockouts, with that reactions being taken out, or the gene knockouts, now I could see the ethanol production is increased. Now it's still the organism grows aerobically. So this is for demonstrational purposes that we show that how you can engineer an organism to optimize the production of certain compounds. Then we import omics data. So here I imported a transcriptomic data set that you can access the same data set that I have imported through these links. And also I list the literature. And then you can compare the expression data with the flux distribution. This is the what I was showing earlier. And you can get this analysis with a push of a button, and see how the model agrees or disagrees compared to the fluxes against the model predictions and the expression data. And similarly, I load metabolomics. I know I'm going a little faster to show the narrative workflow, but you can access this narrative. You can make a copy of this narrative, your own copy, and then run all the apps with your own data. You can play with it. So don't worry about that. That there's lots of content here, and I'm going faster. But at your leisure time, you can explore this content in detail. So I'm here loading a metabolomics data set, and you should be able to see the metabolomics data in a heat map or in a table view, and derive any inferences from that. And finally, you can visualize the metabolomics data and the transcriptomic data on an Asia-based map. In this case, this is the entire yeast metabolism. So this is a big map. That's why the labels are so tiny. So you can increase the size. You could see the glycolysis TCA cycle and the ethanol metabolism area. And you could see where the right now it's showing the fluxes, and you can switch to gene expression, and and you can see where the fluxes are painted, where the gene expression are painted. And also the circles show the metabolomics data, and the color intensity and the size of the circles represents the abundance of the metabolomics. With that, I think that's the end of this workflow. And we have about 5 minutes for questions, and I'm happy to take the questions. And so next, Chris is going to talk about the how the metabolomics get integrated into the models and what you can do with that, all the good stuff coming in about 5 minutes.
Janaka, there are quite a few questions in the chat box.
Okay.
A lot of them are being answered in the Discord, but one of them comment was they requested access to the KBase, but they don't have access to EMSL school.
All right. So quickly going back to the all right. So if you go and if you request the access, I should see that request, and I have my colleague Jose Jose, you can also accept the incoming request. Yeah, I can see, like, just few requests right now. So all right, so they should have the access to the summer school organization. Any other questions? So let me see this code. So I'm just randomly picking some of the question. Is it possible to build your own rich media composition module? Of course. Now at that point, then that rich media become a defined media. So you can have you can build your own custom media in KBase. And so we have a technical term that we use for rich media in KBase called complete media. So just to explain about the complete media is an arbitrary media. So what that does is, if I go to this, back to this workflow, and goes to run fluxes analysis app, and if you don't select a defined media, it defaults to complete media. So the complete media simply means that it's an arbitrary media that it will look for any transporter reactions in the model, and it will submit that media. So it will produce that media for the model that the model has the transporter for. But to answer the question, yes, you can have any custom media. It could be rich media, it could be a defined media. It could be a minimal media. Any Zoom chat questions, Junhon?
Yes, what are those values in the metabolomics data matrix? That's one of the questions. When you show the heat map.
So these are normalized abundance values, and the units can be varies. I on top of my head, I don't remember the units here. So units can be varies from milligrams to kilograms, or whatever the units that the user provides. So that's based on the user input.
Okay. Another question was, can we easily subset and visualize the network? I think it was related to the Azure map.
Say that one more time.
Can we easily subset and visualize the network?
So yes, so if I understand the question, so asking that, can we visualize the network or the part of the network, right? Mm-hmm. So the maps right now, so let me go to the Azure Pathway Viewer. So this is still in beta. So we are experimenting things, and this is a new app. And in the map catalogs, there's tons of apps that we split the biochemistry into different maps. And you can see, like, histidine degradation, TCA cycle. So you have different maps in this map catalog. And that's one way to use the map. And if you have your own Asia map, you can upload those Asia map into the KBase very easily. So using the there's an app called import JSON file as Asia map from staging area. So if you have the JSON version of the map, that you can import your own custom map into the narrative as well.
The related question was, can I upload a model prepared from other softwares into KBase and perform analysis?
Absolutely. So you can use Cobra Toolbox. You can make Cobra models outside, and you can do all the model curation, editing outside, and then you can upload that model as an SBML or as a tab delimited file into the KBase. And then also you can download and you can edit and you can upload back. You can go cycles like that.
And there was a request for a link to the narrative in the chat.
All right. So,
and I believe the rest of the questions are well answered in the Discord.
Okay. All right, so there's no more questions. That I'm going to stop sharing. And so please, if you have any more questions, just type it in Discord and we can all answer your questions. And as also, like in KBase, we are always here. If you want help, just reach us if you need help and how to do analysis, what kind of tools that we have available. And we can help you to do any analysis that you want. If you want to collaborate with us, we are open to collaborations. Just reach out to us.